Let's talk about muscle performance. We also need to talk a little bit more about muscle structure and muscle function. Structure is the parts that make up the whole. Function is what the muscle actually does. In this case, we're talking about muscle. And performance is how well that muscle or a muscle or groups of muscles perform or generate force in essence. So I'm going to go through two key concepts in this presentation regarding muscle performance, and that's force velocity and length tension. And force velocity is simply the relationship that describes that the amount of force a muscle generates is partly a function of how fast it's being asked to generate force. So there's a relationship between how fast and how fast it's being contracted and how much force it can actually generate. The other relationship is the length tension relationship. And that relationship describes the ability of a muscle to generate force is partly a function of how long that muscle is. I always like to start off this lecture with giving a very simple example about that, in that I tell people to, okay, grab two fingers and squeeze as hard as you can. Then I say, go through some wrist extension and then tries to squeeze again. And you'll find that you won't be able to squeeze as hard. You won't be able to generate as much force when you've gone in this position versus this position. And naturally, you start with this position. And same thing, if I go through a lot of wrist flexion, I try to contract. I can't contract, I can't squeeze as hard. Why is that? Well, it's partly because of the length of the muscle in this position versus this position versus this position. And as the muscle gets too long or too short, it doesn't have as great of an ability to generate force. So these are two key concepts we're going to go through in this presentation. Hopefully walk away with that. And the other pieces of information in this presentation you probably have covered in a class like anatomy, physiology, or anatomical kinesiology. So let's go through the presentation. All right, so we're going to go through muscle performance. And as I said, the two key concepts or relationships that you want to walk away with is the force velocity relationship and the length tension relationship. Now, what's important to know is that when we start talking about how to test muscle force, what we are usually doing is talking about two different types of tests that ultimately help us understand muscle performance. And if you're doing reading in this area, it's important to know the difference between these two terms. And the two terms are in vitro and in vivo. When we test a muscle in vitro, what we mean is we're taking that muscle out of the body and testing it in some apparatus. So obviously that would be animal type of muscle. Frog muscle, rabbit muscles are common to use in this type of testing. But we're taking the muscle out of the body and then stimulating it in some way, shape, or form, or doing something with the muscle besides having the brain involved with the contracting the muscle. All right, so that's in vitro. In vivo, we're leaving the muscle in the body and we're doing testing of how much force, and sometimes what we really mean when we're doing in vivo testing is how much muscle torque we're generating about a joint. All right, so now we've got the muscle in the body and we're testing it that way. For most of those types of tests, the in vivo testing, we can't forget that the brain is involved with contracting that muscle. And I say that because there's a lot of interesting work regarding how to generate maximum force of a muscle and factors that influence that ability of that muscle to generate force. An easy one to just sort of put on the table and talk about is motivation. And maybe you're just intrinsically motivated to try as hard as you can to contract that muscle. And maybe you're able to. But a lot of people also benefit from extrinsic motivation, or people yelling, go, 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 something external to the person actually influencing their ability to generate force at the muscle. And so that's important to just recognize, is that the brain is involved in vivo testing. Now you can hook up instruments to the muscle and actually externally stimulate the muscle to contract. But at the end of the day, most of the work that is related to describing length tension and or force velocity relationship. In vivo, it's the person doing the contracting, and you're telling that person, okay, now we want you to contract as hard as possible at this velocity or in this position. Okay, so keep that in mind. There's two types of testing, and I'll even bring this up as we start talking about some of this work as we go through this presentation. Okay, so muscle structure, function, performance. You have had classes on muscle structure, so gross anatomy of a muscle. We know we have a bone, and the muscle connects to the bone via a tendon. We also know ligaments, which is connective tissue, connect bone to bone. But there is also a region, and I should have drawn this on this graph, that is a mixture of muscle contractile units, muscle cells, muscle fibers, and tendon, and we call that the myotendinous junction. All right, so that's in this region here. Then we have the belly of the muscle, or in this case, I'm drawing a fusiform type muscle, and it has all these different groups of muscle fibers that are grouped in fascicles. A muscle fiber is a muscle cell, and when you take biology or any cell type of course, the muscle fiber, which is a muscle cell, has all the units of a typical cell that just organized in a way that allows that muscle cell, muscle fiber, to shorten. And so in the muscle fiber, there's organizations of myofibrils and ultimately myofilaments, the actin and the myosin filaments that interact with each other that allow for the muscle cell to shorten or try to shorten. And I've got sarcomere on here. The sarcomere was within an organization of myofilaments. This is a class that you would have already had anatomy and physiology or anatomical kinesiology. So this I know is a review, so I'm not going over this in depth. An important part of representing or looking at the gross structure of a muscle is to know that these fascicles are surrounded by connective tissue. There's connective tissue at the tendon, there's connective tissue around the muscle fiber, there's connective tissue around the muscle itself. And so there's a lot of connective tissue. The connective tissue is in essence elastic material. And that's important to remember because when we start looking at the ability of a muscle to generate force on a bone, part of that ability is due to the elastic material within the muscle. So we have the contractile element, which is the basic unit of the muscle causing contraction, is the sarcomere. That's part of the structure within the muscle cell or the muscle fiber. Okay, so just keep that structure in the back of our mind. And right now, again, I'm only representing a fusiform muscle, but there's other shapes of muscles as well. Some other muscle characteristics, or some characteristics of muscles, just interesting to know, the human body contains over 400 skeletal muscles, and it's about 40 to 50% of our total body weight. The characteristics of muscle include these: an irritability. That means the muscle can respond to a stimulus. Obviously, that comes from the brain or the central nervous system or some type of reflex type of action. That's an electrical stimulus that ultimately causes the muscle to try to contract or to contract itself. And then the other characteristic is exactly that, contractility. So the muscle can shorten. And extensibility, the muscle can lengthen, but it's always important to remember the muscle can only try to contract. It can resist the extensibility of a muscle, but it does have the ability to get longer. We can pull it and it will get longer. And the other really important characteristic is this elasticity. A lot of what we do in human movement and understanding performance may actually come back to the importance of the elastic ability of the muscle. That means it can return to a resting length. And typically when we start looking at the elastic behavior of the muscle, we're not too concerned about the actual contraction that's happening. In fact, some of the work that we look at with running, we often think the muscle is simply contracting in a way to make the whole muscle tendon unit a particular stiffness in terms of a spring. And so now the muscle is not trying to contract so much as it is just trying to create an appropriate tension So that muscle actually behaves like a particular elastic rubber band, if you will. All right, so we'll get a little bit into that when we start talking about things like running gait. Okay, the function of muscle, there's lots of functions, and a lot of times we like to group these into primary functions and then other things. So the primary function, especially as we start talking about human performance, is to produce movement, to maintain posture, and maybe even to stabilize joints so that other movements can happen within the body. In essence, what we're talking about, the muscle key function, the primary function, is to generate force. Other functions include protection of organs and just protection of structures in the body, and then also regulation of body temperature. So shivering, for example, allows you to generate heat to maintain core temperature if the environment is quite cold. Right, so those are others. When we start talking about analyzing human performance, we're pretty much zeroing in on the ability of muscle to generate force. But there are times that we start looking at other functions of muscle in the body. And I put ... because this list can actually be much longer. Okay, so gave the structure and mentioned the sarcomere. And again, this is information that you would have had in another class. This is just a drawing of the sarcomere, a basic contractile unit of the muscle cell, muscle fiber. And we have our thick filaments, the myosin, and our thin filaments, the actin. And I did not draw additional proteins in here that are represented, that are important to muscle contraction. I just wanted to do a simple model for the purposes of this presentation. And as you know, this myosin has heads on it that reach out to the thin filament, and through the process of the sliding filament theory, that during contraction, during shortening of a muscle, these cross bridges form and pull the thin filament in towards the center of the sarcomere. The filaments do not change length, they simply slide over each other, hence the sliding filament model. And it's called a model because we don't know all the intricate details of the actual contraction, but we know quite a bit about the structures of these proteins as well as this ability to shorten. All right, so keep this in mind because some of this is going to be important as we go along. Okay, functions of muscles. So when we talk about what muscles can do, they can play a role as an agonist, or we would also say prime mover. Those are synonymous. That means that the muscle is contracting in a way that functions to cause the movement. So if I'm looking at throwing, I'm going to look at agonist muscles in certain groups based upon what phase of movement that we're actually in. Or if I'm doing an elbow flexion, I'm going to be looking at the biceps as an agonist type of muscle. Antagonist means it functions to resist movement. All right. So if I use my bicep curl example, my bicep brachii are going to be the agonist. My antagonist would be the muscles that would tend to cause extension. So maybe my tricep. Typically in a bicep flexion, the triceps are very quiet, but it still serves as a role as an antagonist. If it does fire, it's actually resisting the elbow flexion. Those two are pretty common, and I'm sure you're familiar with those. Another one is stabilizer. And this is important in that the muscle or muscles can function to fixate one area so that another movement can happen. It's very common when we start looking at upper body movements, and often we need to stabilize the scapulothoracic joint in a way to allow certain movements about the shoulder, the glenohumeral joint. And likewise, we may need to stabilize the pelvis during running in a particular way in order to allow for hip flexion to occur. Synergist means the muscles function in a way to assist other muscles. So it may not be the prime mover, but it is also assisting in that movement. And the last one is sort of an odd one, but it's called neutralizer. And what we mean by neutralizer is a little different than stabilizer, is that when this muscle contracts, it prevents an undesired movement. An easy example is going back to elbow flexion, the bicep brachii. The bicep brachii not only causes elbow flexion, but it also causes supination. And so if you're going to go through elbow flexion, but you don't want supination, you need to have other muscles fire in a way to prevent that supination from happening. That would mean that they would be neutralizing that supinating action of the bicep brachii. Okay, some other aspects of muscle function. Again, these are terms that you should be familiar with already. Isometric, meaning that the muscle is contracting, but there's no change in length. This is interesting in that when we start looking at in vitro versus in vivo testing, we may actually see something a little different when we start talking about isometric. Typically in isometric in vivo, what we really mean is there's no joint movement, because we aren't really actually measuring the muscle change in length, but we're looking at what's happening at the joint. And I mentioned this for running earlier, is that often we think about running happening as a series of concentric and eccentric contractions of like the quadriceps, the rectus femoris, or what have you. Well, what may really be happening at the micro level, if we looked at the muscle, the actual muscle length, not the muscle tendon length, the muscle during running may actually be contracting isometrically, but simply putting tension on the muscle tendon unit so that it behaves at a particular elastic stiffness. Right, so our eye may say, well, we're going through elbow or knee flexion or extension, and the muscle may need to be contracting that way. But remember, the muscle tendon unit has that elastic behavior as well. And the contractile unit of the muscle may actually only be contracting one way, or that may not seem the same if we pull the muscle out of the body and define muscle shortening, muscle staying the same length, or muscle lengthening in a particular way. When we do get to a testing situation, though, I will refer to a muscle acting isometrically or concentrically or eccentrically, and we will interpret those terms as this right here, that if I say the muscle is acting isometrically, that means there's no change in length of the muscle and no change in length of a particular joint. But if we do a little deeper dive in there, there can be a different interpretation. And then the final, we have isotonic contractions where there is a change in length, and this would be same tension. And really what this means is same external weight. So if I'm holding a 25-pound weight, and I can contract the muscle and have shortening, so I have concentric, or I can contract the muscle and the muscle is actually getting longer. It's not that the muscle is contracting and getting longer, or it's not causing that lengthening, but something's happening. So if I'm holding a 25-pound weight, and I relax the muscle a little bit, it's going to still try to contract and try to shorten, but it's actually going to get longer. There's a torque that's actually causing that elbow extension that's greater than the torque that's causing the elbow flexion. And so the muscle length is increasing during contraction. And then the final grouping of muscle action would be isokinetic. This technically means same force. And I've got a series of presentations on isokinetics because it's a really interesting area to look at and to use for testing of muscle strength and muscle performance. But we often will define this as angular speed is constant during the contraction. And you'll see if you do watch the isokinetic presentations, that angular speed is constant because the tension that's being generated by the torque that's being generated by the muscle is matched by the torque of whatever machine's being used. So that's the same kinetics, if you will, when we start looking at isokinetic. But we typically, if you look this up in a textbook, they typically will define isokinetics as angular speed is constant, which means angular acceleration is going to be 0, and the sum of torques add up to 0, meaning that all the torques are matched. You can do isokinetic contractions concentrically and eccentrically. And when we do this on a machine in our lab, like the Biodex or Cybex or what have you, it does take a little bit for the brain to figure out how to contract the muscle eccentrically while we're going through. If I go back to the bicep, if I'm trying to contract that eccentrically, I'm going to be going through elbow extension. And sometimes it just takes a little bit to learn that movement when we're doing the testing. Okay, muscle performance, how well the muscles do these functions, how well the muscle functions are carried out. And really we're zeroing in on the ability to generate force. Some of the factors that influence muscle performance or the ability to generate force are these right here. I'm not actually going to get into fatigue in this presentation, but angle of attachment, length tension relationship, and force velocity relationship are them. And actually I need to add in pennation angle. Sorry, editing on the fly. All right. Angle of attachment, and I'm not really going to get into fatigue, but this is a really interesting area to study. So angle of attachment and pennation. So angle of attachment is just as it sounds. The muscle via a tendon connects to the bone, and this is the upper arm, this is the forearm, here's the elbow, so the axis rotation. I'm holding an external weight, and I'm looking at the bicep braking. That angle of attachment will ultimately influence the amount of torque that can be generated about this joint. Okay, so that's important because again, when we start doing in vivo testing, we typically are testing the ability of this muscle to generate torque. We're not actually putting transducers in the muscle to figure out how much muscle force is being generated. And we'll go through this when we look at strength for a little deeper. Angle of attachment and is that. So pennation angle is not all the fibers run parallel to the tendon that's connecting to the bone. So here are different types of pennation muscles. So the importance of understanding that angle of pennation ends up influencing how strong that muscle is, as well as how much range of motion or how much it can contract or how much it can extend, because the pennation angle will influence how much shortening can actually happen for a muscle. For generating force, of the pennation of a muscle means that the muscle fibers are running at an angle to the line of pull of the muscle, and you can actually get a stronger muscle because you have more fibers that are generating force along that line of pull of the muscle, but it actually ends up having a less ability to shorten, or how much it can actually shorten. All right, whoops. Okay, so here are the two big relationships that we want to get to. The length tension relationship, as I started this presentation, the amount of force, and this would be in vitro testing, where we take the muscle out of the body and test it. This graph is very characteristic of that type of testing. The amount of force that the muscle can generate is a function of how long that muscle is. 100% represents the resting length of the muscle. So in vitro testing, we take the muscle out of the body, we lie it down on some apparatus, and it comes to resting length. And then that muscle is attached in a way that you can then measure force when you stimulate the muscle. And then they put the muscle at different lengths and stimulate it again and measure the force that way. The reason why that's important to know is that when we're looking at muscles in vivo, we actually don't know what the resting length is. So we would typically talk about an optimum length in vivo. All right, so because we don't know, is anatomical position, is that a resting length? We don't know. That you don't know unless you take the muscle out of the body. So this 100%, when we look at in vivo, would simply be called an optimum length. And it is somewhere in between the maximum shortening of range of motion and the other end of the range of motion. Somewhere in there is an optimum length that the muscle can generate the most amount of force and therefore the most amount of torque. When it's too short, the filaments are butting up against each other at the extreme end, and the cross bridge formation simply cannot bring the filaments together any further. When the muscle's too long, we may actually be losing the ability of cross bridge formation, the number of cross bridges that can be formed, and therefore losing the ability to pull the thin filaments in towards the center of the sarcomere. And again, there's going to be some optimum length when we're dealing with in vivo testing where you have the maximum number of cross bridges that can be formed. All right, the force velocity relationship, as I introduced in the beginning, the amount of force a muscle can generate, and this is in vitro testing, again, the same setup at take the muscle out of the body, put it on an apparatus, stimulate it, but stimulate it in a way that allows shortening at different velocities. And when the muscle is, this is force on the y-axis, this is velocity on the x-axis. And as velocities increase to as the muscle shortens, the ability of the muscle to generate force is reduced. And typically that's because these cross bridge formations have a time that they that needs to happen for the cycling of that cross bridge formation. It has to attach to the thin filament, has to pull the thin filament out and release and then return to its original length. There's a limit to how fast that can happen, and not everything happens all at the same time when muscles are contracting, and so not all cross bridges are being formed at the same time. And so there's a rate limiting factor in terms of how many cross bridges can be formed. And in essence, as the muscle force is going down, we're having reduced ability for cross bridge formation. So faster velocities, less ability to generate force. At zero force, that would be an isometric contraction. Again, when we're talking about in vitro, we definitely mean that there's no lengthening of the muscle. And then on this side is negative velocities, meaning that the muscle is actually being pulled longer while it's trying to contract. And you can see here that the muscle force actually increases, and then it would, in vitro testing, it would increase until at some point there's a failure of the muscle and there's damage, and then the force would drop off. Obviously, we don't do that type of testing in vivo, where we go to a point where we're actually going to have damage to the muscle. Often the brain will shut things down well before there is a threat to the muscle structure. But sometimes the brain doesn't act fast enough or detect that fast enough, and that's where we start having injuries to the muscles due to some eccentric loading. All right, so in vivo, this curve is generally accepted to be the same as in vitro, with an exception that really what we're talking about on this y-axis is torque, and on this axis, angular velocity. And I'll explain this in greater detail in the isokinetic video series. But we are typically testing the ability of a muscle to generate torque about a joint, and as that joint goes through faster angular velocities, that there is less ability for the muscle to generate torque. And that is related to this in vitro relationship between force and velocity. Okay, just to tie this all together, I reviewed muscle structure, which you would have had in other classes, and some characteristics of muscle: irritability, contractility, extensibility, and elasticity. Muscle function can function as an agonist, prime mover, antagonist, a resist movement, stabilizer, stabilizing a particular area for other movements to happen, synergist, assisting an agonist, and neutralizer, preventing an undesired movement that a muscle would cause. And we should have an understanding of what we mean by isometric, concentric, and eccentric contractions, both in vivo and in vitro. In muscle performance, I went over the length tension relationship, the force velocity relationship, and I did leave angle of attachment and pennation. These are really probably a little bit more related to muscle structure, but ultimately they will come back into play when we start looking at how strong a muscle is through a range of motion. And when you go through the other presentations that talk about strength or isokinetics, we'll talk about sticking points within a range of motion of a joint. And that sticking moment is partly due to the angle of attachment, but also partly due to the length tension relationship that we're going through now. Okay, that's it for muscle performance. Thank you.